Today, I'm going to be sharing my senior project, looking at the impact of microbiota on group structure of Drosophila melanogaster. I work in the salts lab, and my graduate student mentor is Eric Weiss. First, let's talk about what the microbiome is and why it's significant. An animal's microbiome describes the sum of its microorganisms, so things like bacteria and viruses. In modern microbiome research, the most commonly talked about is gut bacteria, as that's been implicated in a wide variety of human diseases. Because of its role in human health, microbiome research has surged over the last decade. Fruit flies, Drosophila melanogaster, are an excellent model organism for researching the microbiome because theirs is simple and easy to manipulate. Despite recent interest in it, studying the microbiome using fruit flies is nothing new. All the way back in 1927, Dr. Helen Steinfeld of UC Berkeley performed the first known fly microbiome experiment, in which she found that flies that had their microbial communities removed, also known as exenic flies, had shorter lifespans. In the last 10 years, experiments looking at the microbiome of flies has found that it impacts mate choice, social attraction, life history strategy, foraging, nutrient digestion, and more aspects of physiology, behavior, and life history. Also, things like mate choice and social attraction are important in determining social networks. Because of this connection, I thought it would be interesting to look at how the microbiome may ultimately influence group formation and dynamics. Previous studies have found that social network influences microbiomes. This study is from 2015, and it found that the positioning of baboons within a social network could partially explain differences between their microbiomes. But for my project, I decided to flip this around and look to see if an organism's microbiome might actually influence its social network position, its position within a social network, and the formation of the social network of a group of individuals. So like I said earlier, those things like mate choice and social attraction influence social network formation. And because they can be influenced by the microbiome, I just decided to try to tie these concepts together. So in order to look at this question, I had to rear groups of both exenic, meaning germ-free, and wild-type flies. The ultimate goal was to record social groups of each type and then use social network analysis to look at differences between the formation of each group and also the position of individuals within groups. So the first step of this process was to collect eggs for rearing both the exenic and the wild-type flies. You can see here I have an image of my egg collection apparatus. At the bottom, there's a plate where the flies would lay the majority of their eggs, and then I would collect those eggs using a paintbrush. After that, in order to make the exenic flies, I treated them with a mild bleach solution. The pantyhose actually that you see here on the previous page, so I would place the eggs on there, and then in a sterile hood, I would basically use a dropper to put the mild bleach solution onto the eggs. And then I would also let them soak in that mild bleach solution for 2 minutes. And I repeated that process twice before doing the same thing with distilled water. So once the eggs had been treated with both the mild bleach solution and water, I would add them to sterile autoclaved food. And then they would then develop into exenic larvae and then finally into adults. And this process took a lot of fine tuning, but definitely got it down by the end. So after there were adult exenic and wild type flies, I was able to separate them into 3 different experimental groups. So shortly after the flies emerged, I separated them into groups of males and females to ensure that all flies would be virgins when they would enter the experimental chambers. So there were 3 different groups that I videotaped for this experiment. The first was all wild type flies, in which I had 10 male flies and 10 female flies. Second, all exenic flies. So again, 10 males and 10 females. And then finally, a group that was half wild type flies and half exenic flies. But each of these groups had 20 flies total. In addition, the videos are 20 minutes long, and I have a goal of 10 plus videos for each category that I'm well on the way to. So once the videos are done, and already starting now as well, I'm using these different fly tracking and social network analysis softwares in order to quantify the interactions of flies within the social networks and to understand how the social network formation might differ between the germ-free exenic flies and then the wild-type flies. So my graduate student mentor, Eric Weiss, has done a lot of research using social network analysis and fly tracking. So he's been guiding me along this process. And the goal by the time I graduate is to definitely have the fly tracking done on all the videos and have a good idea of how the analysis will proceed so that I can either be part of that or pass it on to a future undergraduate student. And this is a picture, not from my experiment, but it shows the general setup of what the arenas with the different social groups look like. And then they're videotaped from above, as you can see. So while I haven't been able to analyze the social networks yet, I do have a few predicted outcomes. The first two are actually conflicting, but I'll explain the justification for each. So first, that exenic flies may actually form less stable social groups. I thought this because previous research has found that germ-free exenic flies are less able to distinguish between mates, and they're also generally less interested in spending time in areas with a large number of other flies. And so I was thinking that their groups might ultimately be less cohesive, perhaps have unusual clustering. And so overall, I thought that they would be less stable social groups. However, on the flip side, previous research has found that exenic flies are more likely to follow a live fast, die young kind of life history, in which they lay a lot of eggs early on and then die at an earlier age. So I was thinking that that might cause them to actually have more interactions with other individuals in the group because of an increased tendency to mate a lot early on. And finally, a third predicted outcome is that within the mixed treatment, wild-type flies might be more central in the social group structure than the germ-free exenic individuals. So I'm so excited about exploring this topic because it's hopefully going to help uncover another layer of why social groups form, how they form, and even what the implications of dynamics within a group might be. It's really important to look at social network structure because it helps us to understand processes that are influenced by it, like the communication of both information and disease. And so it's been really cool to be able to take my idea and really dive deep into this question. And so I'm so appreciative to both Dr. Saltz and to Eric for supporting me with this project. It definitely would not have been possible without both of you. And also thank you to my fellow undergraduate researchers who have provided feedback along the way. So thank you so much.